Finally, welcome to the last and fifth part of our discussion on special stains, which will involve the stains for pigmentation, microorganisms, and the other important stains used. Pigmentation is the process by which substances that absorb visible light and produce color are deposited in the body, be it in normal or abnormal conditions. Normal pigmentation is involved in the normal metabolism of the body, such as the color of the skin, while pigmentation produced by disease is called pathologic pigmentation. There are three groups of pigments, namely endogenous, exogenous, and artifact pigments. Endogenous pigments are produced within the tissue to serve a physiological function, or they may be byproducts of normal metabolism. These endogenous pigments are further classified into hematogenous or blood-derived pigments, non-hematogenous pigments such as melanin, lipofuscin, and chromaffin, and endogenous minerals such as iron, calcium, and copper. The following are the different hematogenous or blood-derived pigments. We have hemosiderin, which is the iron-containing pigment of hemoglobin, seen as yellow to brown granule. Next is hematoidin, which is the iron-free pigment of hemoglobin, seen as bright yellow. Hematin, which is hemoglobin without the globin molecule. Hemozoin, which is a black granule formed by malarial parasites. And hemofuscin or lipofuscin, an iron-free brownish-yellow pigment seen in hemochromatosis along with hemosiderin. It is also known as the wear and tear pigment in liver cells. Other pigments include hemoglobin, bile, and porphyrin. Next we have exogenous pigments. These are pigments that consist of foreign materials usually introduced in the body. It is introduced through air, food, medication, or injections. Some examples include tattoo, asbestos, silica, iron, and silver. Carbon is the most common exogenous pigment. It appears as jet black deposits in the lungs, especially of chronic smokers. It may also be confused with melanin, and to differentiate the two, melanin can be dissolved by bleaching agents, while carbon cannot. Some important terminologies are anthracosis and coal workers' pneumoconiosis. Anthracosis is the black pigmentation of the lungs due to large deposits of carbon, while coal workers' pneumoconiosis is caused by inhalation of silica associated with coal and other minerals. Lastly is the artifact pigments, which are simply deposits of artifactually produced materials caused by interaction between components of some tissues and chemical substances, such as formalin. Let us now proceed to the different stains for tissue pigments and deposits. First is the Pearl's Prussian blue and Turnbull's blue. These are both used for staining hemosiderin. The only difference is that Pearl's Prussian blue is for ferric iron, while Turnbull's blue is for ferrous iron. With this being said, Turnbull's blue is rarely used because ferrous iron is less common compared with ferric iron. Next is the modified Fauchet's technique, which is used for the demonstration of liver bile pigments. The Mellin technique is used for bile and hematoidin. This method is considered diagnostic for bile pigments, but produces temporary results. Next is the Gomori's aldehyde fusion and Mallory's fusion stain. It is used for lipofuscin and hemofuscin respectively. And the Mason-Fontana technique is used for the demonstration of melanin and argentafin granules. Mason-Fontana technique has an end color of black. Next is the calcium dilake reaction. This is used for insoluble calcium salts, such as calcium phosphate and calcium carbonate. This method is specifically recommended for staining the skeletal system of embryos and fetuses. The calcium dilate method makes use of either the routine H&E method or the sodium alizarin red S. The routine H&E method stains calcium salts as deep purplish blue, while the sodium alizarin red S stains it orange red. Next is the von Cosa silver nitrate method used for the demonstration of calcium. And lastly, a recall board exam question is the Lindquist's modified rhodamine technique used for the demonstration of copper. In this technique, apathies media is the preferred mounting medium to use. We now proceed to the microorganisms. The microscopic appearance of stained tissues may reveal infection with the presence of inflammatory cells as well as different microorganisms. The stains for a wide variety of microorganisms are as follow. We have, of course, stains for bacteria, such as the Gram stain and the Brown and Bren method. Gram stain differentiates gram-positive and gram-negative bacteria depending on the thickness of the bacteria's peptidoglycan layer. This will be further discussed in your bacteriology class, but what you need to remember is that gram-positive bacteria are stained purple, while gram-negative ones are stained red. The Brown and Bren method, aside from bacteria, is also used for Nocardia and Actinomyces species. Next is the Ziehl-Neelsen method, which is used for acid-fast bacteria. It is recommended for Mycobacterium species, an example of which is the Mycobacterium tuberculosis, one of the most common acid-fast bacteria and the causative agent of pulmonary tuberculosis. The weight-fight technique is used for the demonstration of the causative agent of leprosy, also under the Mycobacterium species, which is the Mycobacterium leprae. And aside from that, it is used for the demonstration of Nocardia species as well. Auramine-rhodamine stain is a fluorescent method used for the demonstration of Mycobacteria species. Toluidine blue is for demonstrating Helicobacter pylori, the bacteria that causes gastritis. Next is the Dieterle method. This is used for demonstrating spirochetes and especially Legionella pneumophila. Legionella is the causative agent of Legionnaire's disease and is known to be spread through air conditioning units. Next are the different stains for spirochetes. We have Levaditis method, modified Steiner and Steiner technique, and the Warthin-Starry method. For the modified Steiner and Steiner method, the solution is milky, so it must be used immediately. This method also stains Donovan bodies, fungi, and other bacteria. The Grocott-methamine silver or the GMS method is for the demonstration of fungi. PAS or the periodic acid shift may also be used for the demonstration of hyphal elements and yeast. Lendrum's floxin tartrazine is used for the demonstration of viral inclusions. Orsain is for the hepatitis B surface antigen or the HBsAg. And lastly is the Giemsa stain, which can also be used for blood and marrow parasites such as Leishmania, malaria, and trypanosomes. Also, it demonstrates toxoplasma, spirochetes, and other bacteria. That concludes our stains for microorganisms. The next slides are the other important stains. Some have already been discussed previously in the recent parts, so they may seem familiar to you already. So we have the Van Gieson, if you will remember, it is for collagen, which is a combination of acid fusion and picric acid. Acridine red 3B for calcium salts and sites of phosphatase activities. If you will remember, Alcian blue, and also we have aniline blue for counterstaining of epithelial sections and also for collagen, reticulin, and others. The basic fuchsin, which is used for deep staining of acid-fast organisms, mitochondria, and for the differentiation of smooth muscles. Bismarck brown is another important stain, which is used as a contrast stain for Graham's technique, in acid-fast technique, and in Papp's method. And also, an important information is that Bismarck brown is also used for staining diphtheria organisms. Carmine is combined with aluminum chloride to stain glycogen. Celestine blue, which is resistant to strong acid dyes. It is recommended for routine staining of fixed sections. We already discussed Congo red and crystal violet. Crystal violet has a subtype of gentian violet, which is just a mixture of crystal violet, methyl violet, and dextrine. Crystal violet is used to demonstrate amyloid in frozen sections and platelets in blood. Gold sublimate is used for metallic impregnation, which is made up of gold chloride and mercuric chloride. We have already discussed iodine as well, which is considered the oldest stain originally used to demonstrate starch granules. We have subtypes of iodine, which are Graham's iodine and Lugol's iodine. The next one is a favorite board exam question, which is Janus green B, used to demonstrate mitochondria. Malachite green is encountered in the parasitology lab for the demonstration of Ascaris eggs, as well as red blood cells, and it is also known to be a bacterial spore stain. We have methyl green, methylene blue, methylene violet, neutral red, night blue, or cyan, which we already mentioned as highly recommended for dermatological studies, osmium tetroxide, picric acid, Prussian blue, which is used for the manufacture of paint, rhodamine B, silver nitrate, toluidine blue, and lastly, Victoria blue, which is used for the demonstration of neuroglia in frozen sections. So that ends our discussion for special states. If you have any questions or clarifications, drop them on our forum discussion. Thank you for listening and God bless.